Back, and we are discussing the our previous chapter, chapter number 12, and we are continuing where we left off. So we are discussing about the composition of the blood cell, a whole blood. So as we know, after centrifugation we receive three different layers. One is plasma, and the middle one is buffy coat, which include platelets and leukocytes, and then we have neutrophils, lymphocytes, monocytes, eosinophils, and basophils. And in erythrocytes, we know we have erythrocytes, which are red blood cells. So today we're going to discuss this erythrocytes, and then we're going to go to this buffy coat, and the last one I'm going to discuss about the plasma. So it's going to be in the separate video. So first we're going to discuss about the erythrocytes. Let's start. So, as we know that if we have to study blood cell in histology, so we have to take a blood and we have to put it on the microscopic slide, a drop of blood on the microscopic slides, and we have to put some dyes on it to study about the structure of the red blood cell, what inside in the red blood cell. So first we do is to prepare the blood smear. We have to withdraw the blood from the finger, and then we place that drop of blood on the slides and then put stains on it. We also learn different types of stain that we use to study red blood cell. So and then after the blood dries, we apply the stain, and then we rise rinse, sorry, and then we place a cover slip on top, and then we put that slide onto the microscope stage, and then we can see this type of structure inside the microscope. We see the purplish and pinkish structures. So let's start. So observation of their nuclear and cytoplasmic characteristics. Blood smear are routinely stained with mixtures of acidic is erosin and basic methyl blue dyes. These two dyes mix together to study the characteristics of cell of red blood cell. These mixture may also contain dyes called azures, that are more useful in staining cytoplasmic granules containing charged protein and proteoglycans. So basically, in this paragraph, right, telling us about the different types of stains we use to study different cytoplasmic structures, such as it explained that if we need to study some charged protein, some proteoglycans, so we use different dyes, which is that name is azures. We also use a mixture of erosins and basic dyes. There's also the azerophilic granules produce metachromosomal stained leukocytes that's seen with mast cells in connective tissue. So this dye is used in the connective tissue to see the mast cell. Some of these special stains, such as gamismas and white stain, are named after hematologists who introduced their own modification into the original mixture. So basically, these are the two stains that are also used to study the blood cells, and some of them are modified by histologists, few histologists. So let's continue. Erythrocytes. Erythrocytes are red blood cells. What is the important in the structure? So if we see in the microscope and we see erythrocyte, we can see they don't have nucleus and completely filled with oxygen carrying protein called hemoglobin. So RBCs are the only blood cell whose functioning does not require them to leave the vesiculture. So this is one of the important um, importance of the red blood cell. Human erythrocytes suspended in an isotonic medium are flexible biconcave discs. They are approximately 7.5 in diameter. Is thickness is described in here. I'm not going to read this paragraph because it's very easy. So I'm highlighted in the yellow what are important things I need to study. So the biconcave shape provides a large surface to the volume ratio and facilitate gas exchange. As you know, red blood cell is oxygen carrying molecules, and it transfer oxygen in different or to the different organs. So erythrocytes are normally quite flexible, which permits them to bend and adapt to a small diameter and irregular tons of capillaries. As you know, some of our veins, arteries, capillaries, some of them are big, some of them are small. So this is the structure of the one red blood cell is thickness, and its thickness from the center and the thickness from the corners are described in this picture, in this sectional view. And this is the microscopic slide picture of it. Observation in the vivo show that a angle of capillary bifurcation. So capillary bifurcation means that one capillary is dividing into branches. Bifurcation means division of branches, or for example a tree has different branches like that. So bifurcation is the branches. Erythrocytes with the normal adult hemoglobin frequently assume a cup-like shape. In large blood vessels, RBC may adhere to one another loosely in stack called rulax. What is rulax? So basically, rulax are the red blood cell in the line, like 1, 2, 3, 4, like in this picture. It shows when it's like little bit of bend, the RBCs get in the line to go through that area. So rulax is the in the small vessels, red blood cells often stack up in loose aggregates called rulax. The standard size of RBC allow one to estimate that the vessel seen in approximately 15 mm in the diameter. So rulax is the as you see, the vessel is passing through the capillaries or small vessels or whatever the structure is. So this stack-like structure form is loose. So this structure of red blood cell called rulax. So next we're gonna learn about the erythrocytes plasmalemma. What is the surface layer, or you can say lipid bilayer, as in the cell membrane. So erythrocyte have a cell membrane which is called plasmalemma, because of its already availability is the best known membrane of any cell. It consists of about 40% lipids, 10% carbohydrates, and 50% protein. Most of the latter are integral membrane protein, including ion channels, the anionic transporter called band 3 protein, and glycophorin A. The glyco stellated extracellular domain of the latter protein include antigenic sites that forms the basic for the ABO blood typing system. Several peripheral proteins are associated with the inner surface of the membrane, including um, spectrin dimers, of which form a lattice bonds to underlying actin filaments, and chirin, which anchors the spectrin lattice to the glycophorin and band 3 protein. This submembranous meshwork stabilizes the membrane, maintains the cell shape and provide the cell elasticity requires for the passage through the capillaries. Erythrocyte cytoplasm lack all organelles but is densely filled with hemoglobin, the tetrameric O2 carrying protein that accounts for the cell uniform acidophilia. When combined with O2 or CO2, hemoglobin forms oxyhemoglobin or carbaminohemoglobin respectively. The reversibility of these combination is the basics for the protein's gas transporting capacity. Erythrocytes undergo terminal differentiation, which include the loss of the nucleus and organelles short before the cells are released by the bone marrow into the circulation. Lacking mitochondria, erythrocytes rely on aerobic glycolysis for the minimal energy needs. Lacking nuclei, they cannot replace a defensive protein. Human erythrocytes normally survive in the circulation for about 120 days. By this time, defect in the membrane cytoskeleton lattice or ion transport system begins to produce swelling or other shape abnormalities as well as change in the cell surface oligosaccharides complexes. Sequence or worn out RBCs display such changes are recognized and removed from the circulation mainly by the macrophages of spleen, liver, and bone marrows. So here in this paragraph, we have seen the erythrocytes plasmolemma, what they have on the outer surface of the plasmolemma, what they have inner surface of the plasmolemma, what is in the erythrocyte cytoplasm. We have seen this, and we have seen what type of respiration to done by the erythrocytes. We have seen how the red blood cells, the lifespan of red blood cells, and how their lifespan affect on the different structures and different things, and how the macrophages of the from the spleen, liver, and bone marrow remove these dead RBCs from our blood. Thank you very much. We will continue next topic, leukocyte. Stay tuned.